I'd like you to introduce all to with our first session chair, Dr. N.K. Jain, and our first keynote speaker, Dr. Anil Kumar Mishra, to our esteemed audience. I would like to invite Dr. N.K. Jain on the stage. I would request Dr. Rizwan Khan, Principal Skyper, to greet Dr. N.K. Jain with the bouquet. Dr. Jain is an internationally renowned academician and researcher and a senior professor in India who superannuated in June 2014 after rendering more than 40 years of dedicated and distinguished service as a teacher and 25 years as full professor. Starting his academic career in 1973, he worked as lecturer in Pharmacy Holkar College, Indore, reader in Pharmaceutical Technology, MS University, Baroda, professor and principal College of Pharmacy, Nasik, professor and head Department of Pharmaceutical Sciences in Dr. H.S. Go University, Sagar, principal at College of Pharmacy, New Delhi, visiting professor, Dubai Pharmacy College, Dubai, UAE, and Head Pharmacy Department, Delhi University. Subsequent to his superannuation, Professor Jain rendered his service as Director, ISF College of Pharmacy, Moga, Punjab, as Emeritus Fellow, UGC at Rajiv Gandhi Technological University, Bhopal, MP, India. Professor Jain is the author of 24 celebrated books and has contributed over 60 chapters in series on controlled and novel drug delivery system and has contributed over 60 chapters in national and international books. He has to his credit over 485 publications in reputed pharmaceutical journals and has supervised 54 PhD and 142 M.Pharm candidates. He has been a reviewer for several international and national research journals. He has also translated Professional Pharmacy Part 1 by Emil Shroff in Hindi on behalf of Central Hindi Directorate published by Bihar Granth Academy, Patna in 1975. Now I would request Dr. N.K. Jain sir to throw light on the topic and invite Dr. Anil Kumar Mishra on the stage.
Good morning to all of you. It's my pleasure to be here to introduce a renowned speaker, a great scientist, a simple man, and a very good human being. I'm referring to Dr. A.K. Mishra, who is sitting here. Dr. Mishra is working as a scientist at History of Nuclear Medicine and Allied Sciences, DRDO, Ministry of Defense, Government of India. He obtained his PhD in chemistry in 1988 from Varanasi, that is the Banaras Hindu University. During his career, he had positions at postdoctoral fellow at University de Bourgogne, France. He has been a research scientist at University of California, USA, senior scientist at INSERMU 463, France, and visiting professor at Max Planck Institute, Germany. He has published more than 284 papers in various journals of international repute, and he was granted US, European, and Indian patents, several ones. He received several awards, and most prestigious one is the DRDO Young Scientist by Prime Minister Shri Atal Bihari Vajpayee of India in 1999. Currently, he is working in the field of radiochemistry, coordination chemistry, synthetic chemistry, and bioconjugate chemistry. Ladies and gentlemen, the topic which is going to refer to in his discussion, this is a very contemporary topic. And to my students, to all of these students, this maybe is something new so far as their knowledge is concerned, yet it is a very contemporary topic, and in due course of time, during your life as a career, as a pharmacist and as a scientist, you are going to use these things. I do remember that we used these facilities for some of our PhD students, and as a student, as a pharmacy student, you might need those facilities. The topic which he is referring to is related to something which is today as well as the future. And that is related to the real-time imaging, real-time diagnosis, and real-time treatment. That is what now is reflected in terms of another term, that is the theranostics, the therapeutic and the diagnostic systems which are going to take place in the near future. Like you can have something which is nano material, nano size, and at the same time it can help us in diagnosing the disease by imaging and can help in real-time monitoring what is really happening to a drug which is administered to the body, and by that we will know what is really happening in the body with the drug and how much time it is going to take cure or whether it is going to cure or not. These are some of the issues which are boggling the minds of the scientists, and Dr. Mishra is going to throw some light on these things. One important aspect of this topic that is related to the copper, that is the Cu64, which is a requirement of many of the cancer cells, and that nuclide can be exploited. Another aspect that could be utilized that is related to the use of materials, the carbon materials like say quantum dots, like say carbon nanotubes, and sometimes the hybrid of the two, that is the carbon quantum dots, etc. So these are the issues which are going to be discovered, and they will be very much relevant to all of us. And I request Dr. Mishra to please enlighten us with the topic on this very contemporary issue so far as the human requirement is concerned. Thank you very much. Dr. Mishra.
Thank you very much, sir, for a kind introduction. Thanks for inviting me to share that what we do at our institute. Then also the student that can get benefited, that how they have to think to design something, to be very precise, because today we talk about precision medicine, and then to do the precision medicine, we have to be very precise to even design the molecule, which are very useful for doing, like he was talking about theranostic, to having a twin pairs of system that they can be used for diagnostic application as well as therapeutic application. So if you can see is at my first slide that I have put in such a way that if you can see it, you can treat it. But how you can see it, where it is located, how deep it is located, which modalities and which tools can offer to see it. So we do not have many. We have got only 3 types of system that we can see and we can treat it. Either we can really find out where it is located. So we always try to correlate with electromagnetic radiation, because all the tools that we talk about, that completely designed on the basis of electromagnetic radiation. So it is your wish list that which type of tools that you want to use for which application. Here I am talking about radioligands. It means that I will be talking with the shorter wavelength with a very high energy that matters to me. Keeping this in view, that I thought that if I am able to see how I can see it, and then also I have to be very precise. So do I have many options? Either I have a very limited option. So keeping this, I put one slide just to share with you that have a couple of case studies. This case study talks about that anything that happens, how it happens, and what time scale it happens. So this all 3 answers we get from the chemistry point of view, from tools point of view, from precision point of view. So if you look about any metabolic pathway that we want to really map, either to see, then we try to see that how I can do the isotopic labeling to know that this pathway is taking place to make the neurotransmitter, either this pathway is taking place to do the protein synthesis, either any macromolecules that we talk about. But we should not forget about the case studies too, that who is going to drive it to happen this reaction, because we have to have the catalytic pathway. So then it comes about the metalloenzymes, and they are very interesting to drive even very minute reaction and a very instantaneous time scale to see that reaction happens. When reaction happens also we have to determine the kinetics. And to determine the kinetics, again we have to see that how we can see that what time scale it is happening. Can we alter it to really see that our instrument can see it or not? And that's why you can see over here is that we have got the optical imager, we have got the nuclear medicine, and we have got the NMR that we talk about, magnetic resonance imaging, where we talk about T1 and T2 to exploit the property that how tissue they behave. For me, tissue means how molecules they behave, how their parameter is changing. This is very important for me. So what I have done is that I have taken a cartoon here, that if anything I have to exploit in the case studies 1 and 2, I have to be very fast because I want to work with some of the radionuclides which half-life is very short. To introduce you, slowly and slowly I will bring it, that we have to see that what type of methodology I am going to adopt to do the reaction. How long it will take and who will drive it, that reaction should take place. And it is also very important for me that this reaction when taking place to conjugate something, they can be metabolized, they can be stayed for longer time, either they will be killed by some of the enzymes. So also we have to keep in view whenever we're designing this. Once you are very good in methodology, where it comes your intellectual property right, your educational growth, that you gives to the many people for the country, and finally you get translated it for what we say that is a prototype or a preclinical in terms that we always talk about. So even if you look at this picture, the prototype that I'm talking about, I have an experimental model, and this experimental model I would like to see that whatever I have grafted, it really I'm going to see or I'm not going to see it. Once if you do this process of prototype, directly it can get translated for clinical application. The images that you have been seeing, that you can really offer you for translational research. So you can say that we started from the bench, we go to the bedside, and that is what happens when we do the clinical studies. Either when we think about designing any translational research work for different type of disease that you want to target, either it is oncology, either in neurology, either infection that we have been talking. And that's why I have given this radiomolecular medicine and the medicine of tomorrow, either I will say ahead of tomorrow, that we can witness slowly and slowly. I took this cartoon again to explain that you look about the entire organ that you want to see it, and it's being practiced every day. Any type of your renal function is not there, your pancreas is not functioning, your brain is having some problem, your bone is something has happened. For each and everything that you have got the medicine that is called nuclear medicine to see it. But being a scientist, being a researcher, being a pharmacist, working in this area, we are never happy because we wanted to be very precise. We wanted to really see the pathway that has been revealed, either the taking place to design the molecule, which will be very precise and very essential for us. What I will be doing is that I will be taking a couple of examples that how do we design the classical, then more advanced, and then become very specific and precise. And then we'll conclude the talk that the way I have designed it. So look about anything that it starts from the first generation till today, and I'm thinking about tomorrow. So tomorrow also we'll discuss with this slide. So let's see that couple of slides that I have taken. So in this, anything that you want to visualize, just take one example of bone that we wanted to do. So very nicely you can design, knowing the fact by inorganic chemistry, either organic chemistry, what is the complexation constant, what is the stability constant, to know that this is going to bind with this magnitude and it will not be dissociated under physiological condition. So whenever we design something, also we take many parameters together, because enzymes, protein, nucleotides, lipid, they also behave in the similar manner to take whatever you are going to inject. So you look at this example that we wanted to really the bone metastatic lesion, and we designed very nicely with the cyclohexylamine to introduce many denticity to wrap the metal ion very strongly under physiological condition. And then you can see very nicely that where metastatic lesions are located, and you can visualize it. There are difference between these two images that you can see on the top and the bottom. Reason behind is that whenever you want to see, also would like to treat it. So you can see that the technetium that we take, that binds very strongly. But if I want to take something from lanthanides, either A block elements, either I want to take some transition metal, then I have to really increase the density to form a very high coordinating, well-defined radiochemistry, and then that doesn't get dissociated under physiological condition, again then when you talk about, and then you can really load with lutetium, samarium, any beta negative that you are thinking about, and then you can really treat very nicely. So this is the one example that I wanted to share, and then we also took one amino acid that I wanted to flash it, that we produce and we are using in clinic, that was designed just for the protein synthesis based on methionine, and this molecule is being used for breast imaging, a different type of, you can talk the HER receptor, either you can to the PR receptor, either you can think about triple negative also. You can visualize very nicely with this product because it has got a different type of co-ligand which binds very specifically with the nature. So that I wanted to share that how we design this molecule, and then finally we conclude to this one. When we talk about the molecular imaging, that we try to see that any pathway that you want to reveal it. For example, if we talk about the GLUT receptor, and then certainly you have to see what is the precursor, where I can do the radiochemistry to see it. And very nicely, most of the nuclear medicine center and the PET imaging all over the world, they use fluorodeoxyglucose, which is level with fluorine 18, to do the imaging. Very nicely, you can see the image that you are able to. But sometimes it's become background is very high, so you cannot really discriminate between the background as well as where metastatic lesion is located. Then again, we try to see that some of the amino acid you can label. That is, we talk about tyrosine, either methionine that I was talking about. Then you can go again for any nucleosides, either any nucleotides that you want to see that takes part in the cell proliferation, and very nicely you can visualize. So you can see these three images very nicely, that we are able to design a molecular medicine that can go and you can see the images non-invasively without opening, either without doing anything. So this is the beauty of the system that we talk about. I will just take a couple of examples that what we do with this one and with this center, being at the Institute of Nuclear Medicine and Allied Sciences. You can see that when you look at the entire canvas, you have so many molecules for the different type of receptor that you want to really visualize, and you can see very nicely. Why I wanted to show this one is that with these molecules that I designed at my center, I almost generate 10 crores of rupees every year by distributing all over the country. So this is the beauty of the system. This is the beauty of radioligands that we talk about. If you design, certainly you can have to different hospitals you can supply. You can really see that the people are diagnosed properly, not with the only one molecule and all type of disease that you want to visualize. This is our duty to see that which type of molecule has to be injected for the type of tumor, either oncological manifestation that you want to see it. So I would like to take a couple of examples in this, that how we do it. We try to produce the isotope. For that you have to have the cyclotron, and cyclotron is just like sweeping the street or the road when dust comes out to interact with your nostril, and then you start sneezing. In the similar manner, what we do is that we accelerate the particle, and we accelerate the particle in such a way that to perturb the N by P ratio of any nucleides, and then finally it becomes the unstable, and that unstable we try to harbor, and then we say that we have produced the isotope that can be utilized for the different application, keeping in mind that what is the half-life, and then you can see very nicely preclinical, and then finally it goes for the clinical application. So what we do is that we shoot the proton beam, we shoot the deuteron beam, we shoot the alpha beam, and then we perturb the N by P ratio to produce the isotope for our application. I thought that I will take only one example that how we do it. The reason behind is that I took only one example to show that how you can think and design of a molecule that was being used for most of the time in vitro, and limitation with the optical imaging is that if you cannot have the up conversion of the photon, and if the tumor is deep seated in the brain either somewhere, you cannot visualize it, because you do not have any dyes, you do not have any optical probes, which extinction coefficient is very high to see it by any methodology that exists for the optical imaging. And that's why optical imagers are very powerful in ex vivo condition, and by taking the lead from that, we always think that how I can convert this molecule with a radioisotope that can be utilized to really see in vivo what is happening. When I was reading this paper, I got this paper by Magavi et al. That they have very nicely exploited for the sum of the cognition and neuroimaging. And then we thought that, okay, this is very interesting molecule. Besides the onco, we can go for the neuroimaging. But I will just share that how we designed this molecule for very nice way. So Pia was talking about when she was doing her PhD program, either she was doing her chemistry in the very long time back, always she wanted to design the molecule that based on nucleosides to have the different type of precursor to make the molecule very powerful. So what we try to do is that we try to play with the 3', 5' in such a way that we do not alter its biological property. Also we have to see that the molecule that I am playing, it should not be destroyed its property, either its beauty, otherwise you will be nowhere to see that what is happening. So we try to put a partner, and that partner is very reactive in the manner that you can see that we have introduced a alkyne group, and just we do the reaction like we say that click reaction that was developed by Sharpley at long time back, and then you can make this molecule in very nice in the fraction of millisecond, either maximum 2 minutes time you can produce these nucleides. And then having 20 minutes of half-life of this carbon 11, you can do the chemistry as much you want because you have developed a protocol to do the reaction to conjugate the tracer moiety, either the radioisotope that can be very tools for you to guide it. It means that it becomes a very beautiful treasure to see it. So what we do is that we do many kind of reaction condition, changing the different type of R group to satisfy ourselves that we are very good chemists and we can introduce whatever we want. But end of the day, that what is important for us is that to see that we are able to do a partner that does the click chemistry, and then finally we are able to see the tumor where it is proliferating. This is the one objective. So once you are able to see it, then they just go for the chemotherapy, either radiotherapy, either targeted therapy, whatever the therapy they do. But also we have to see that whenever I am subjecting someone for therapeutic application, I am able to really see that is regression is taking place or not. Either the reaction that I am doing, either the treatment that I am giving is really beneficial to the patient, either not. So that also again we have to do the follow-up. So let's see that what we have got here. So if you take the gradient after treatment of many cycles, they say that, okay, I have given the 6 cycles, 5 cycles, 3 cycles of chemotherapy, and if it is not responding, then there is no question of giving the treatment. But you can see this system, that starting from there, you are very nicely able to see that the treatment is really powerful, and then tumor is regressing, either getting to the complete remission that we talk about. And then this is the very nice case that when we can stop the chemo cycle, either chemotherapy, if we see that all the tumors are regressed, either there is no hot spot that we want to see by the tools and technology that we have utilized for the application. So this is the one very important, that how you have to design the molecule to see, to follow up, and then finally we say that we have done a complete canvas in this to show that it works very nicely. But also you have to see a case where complete remission is taking after 2 weeks, where you give for several months this chemotherapy, but there is another case where they say that we give the treatment but it's not responding. Then your treatment planning should be changed. And this is very essential for us, that whatever we are developing, we should have knowledge that this molecule is really useful for this type of clinical outcome, or that is not useful. And then we have to take a call, because we have really beautiful molecules to really see it, and depending upon the type of receptor, which is very essential to right now, to really titrate it and to see that it is upregulated and downregulated by different modalities that we have got. And then end of the day, you can say that this molecule is not going to give any response with the chemo, so they have to change the treatment modalities. So taking this one, we thought that I will throw some light on the few of the other things that you can do. So this was the one example. You talk in oncology, you talk in neurology, you talk the tracer based on vitamin, amino acid, peptides, nucleosides. So now the selection of radionuclides that you have got, you can really introduce, and you can do the type of reaction that I was suggesting to increase the affinity, to enhance the affinity as well as specificity, because you want to be as minimum dose that you want to deliver to see that you are able to do that. So we did a lot of chemistry in this one with the carbon, nitrogen, you talk about fluorine, you talk about technetium. So all this chemistry we do quite often at our institute for the clinical application. So since that technology is getting advanced now, we are getting the hybrid technology, hyphenated technology. Then the clinician, they ask that, okay, I'm able to see by one methodology, but where is the second methodology? Because your molecule is not going to be very beneficial for that. So we have to certainly think, being a chemist, either a researcher, that how we can design that we can ahead of even the tools design, either tools development. So just I would like to share one of the example. You can see very nicely that we are able to introduce fluorine 18. At the same time, we have got the SKICON2023 NMR. So very nicely you can see the brain ventricles flash very nicely, as well as at the same time you can do the 29N silicon what you say that NMR spectroscopy that is not moving this file. So it becomes a really dual probe for us. So what I can do is that I can really see by two ways, and I will give you a very beautiful example for this one. Just look at this picture. I like this cat because this cat has got two type of eyes. So always I ask a question that why it has to have the two type of eyes, and then the question comes out that when I'm able to see by nuclear medicine, I'm able to see by the PET, I'm able to see by optical, many things. But the question comes that having the two types of eyes, that it gives me advantage when I design my probe to see that it makes a high specificity and high resolution to really conclude something for the clinician, that where knife has to go, where surgery has to take place. So it gives me a very good canvas on this one. And then what we try to see, that the example that I was showing in the beginning, you just look at the panel. The first one is the MRI, second one is the PET image, and you look at the third one. So you know that where is the maximum activity and that person to be taken out, because that is the viable tumor. You cannot really distinguish in the radiology. Only radiologists can read it, but for us it is very difficult. But in PET also we can read it, there is a hot spot, but where is the maximum accumulation? Then you can look at the last panel where you can see the very yellow contrast. It means the tumors are aggressive, they are multiplying, they are viable, and they have to be removed. And that's why we always say that once you have got two technology, it becomes 1 + 1 = 3, because to get some information which is very essential for the treatment point of view and the treatment application. So this is the technology that we talk about, the molecular medicine. We talk about the first generation medicine. Now we have to move a little bit that why we have to leave this to one, and we have to think about today or tomorrow to see that how we can design the molecule, which is very specific. Whatever I want, I can do. I can control the chemistry. I can do the conjugation. I can play with it. I can be very precise. All these questions we ask, and then we look way forward, where we want to really see that even a small fluctuation of any receptor at single molecule level, if we can track it, then certainly we can treat it. But how it has to be done? Then again it comes out that you have to have the different type of accelerator. Again, we talk about production of isotope, and you must be knowing that your city is very powerful. I don't know how many of you know that. We have a synchrotron in this city, that is CAT, but that is very giga electron volt powerful synchrotron that you have got. If you want to produce the isotope, it will be Herculean task to separate the isotope once you irradiate it, because the energy is so high, you cannot shunt it, you cannot kill it, and that's why whenever you design for the future something, also you have to take in account that how we can design the system to produce the isotope. I will take a couple of examples that Dr. Jain was also talking about. The copper 64 is a beautiful element in the periodic table for many regions, even to teach the life. Always we say that there is something exception in periodic table. What is the exception? Copper. They never follow the rules of periodic table because they want to make other happy, so they donate the electron, become half filled, either zero. Above principle, when you remember when you were doing your chemistry. So we talk about the scandium, copper, zirconium to produce it very nicely. how you can produce it, you can tell me the time also, that whenever you feel like. So it's, for me is that moving from this to this radionuclide to see the resolution, that what resolution I can obtain. And that's where we try to always change the isotope to have the better resolution in the sub-millimeter level even, and then after that to do the chemistry, to be very specific and precise to do that. So we have got different type of targetry. We are always very essential, because not many people, they have got the liquid, gas and solid type of target material, either targetry where you can do the radiation to irradiation to produce the isotope. So I took this one example for two regions. One region is this image that you see is the gallium-68. That is in the periodic table. Quite often we use this radionuclide for neuroendocrine tumor. There are many other things also we do it. But just see the difference between this image and scandium-44. You are able to really see the micrometastatic lesion, because the resolution is so high that we are not able to see with the gallium. That's why the two regions I have proposed. One is that half-life, so I can cover entire part of the region that where I have been working. Then other person then also can take about the other region that they have been working. And then the beauty of the half-life, that one thing that can be from 68 minutes to 3 hours, so you can distribute anywhere. And then resolution is very visible, that you can visualize very nicely that what is the impact of this isotope when you produce it. But once you produce this isotope, a lot of chemistry has to be done. And the chemistry has to be done in such a way that you do not compromise many things. So what we do is that we try to keep a case to wrap the metal ion very strongly again in the same manner, increasing the density. We try to introduce the metabolizable linker that we don't want to be for the longer time, so it is digested by the enzyme. Then we can do very quickly click chemistry to introduce the recognition moiety. And then to cross the blood-brain barrier, we have to do the lipidization. So also you can cross the blood-brain barrier. So we try to make a molecule that is called the cocktail. So you have got the hybrid molecule, and this hybrid molecule takes care about many things. So you can introduce the recognition moiety, you can do the lipidization, and you can also introduce the recognition moiety. Let's see that couple of examples that in this one, that how we design it. So you just look at this, don't go on this structure. Is a peptide bombacine that is for brain and as well as prostate cancer. We try to see that how we can level with the different radionuclides, that is for the spec, that is with a single photon emitted computed tomography, that most of the time you guys from the pharmacy, we are working almost 7 to 9 years before from all the pharmaceutical institute of this country. But now we are graduated, we have the beta positive, we have got the beta negative, we have got the different OJ electrons to work upon. So what we do is that the same chemistry in a very nice way. The catch is here is that why I have pressed this molecule, that sometime when you do the chemistry and you took a molecule which is antagonist in nature, agonist in nature, co-ligand in nature, but when you do the conjugation chemistry, you change the property. So if you become antagonist, agonist become antagonist, that is very difficult for us to survive with this one. And that's why we try to see that molecules remains molecule, chemistry remains chemistry that we want to see, and also to achieve the specific activity. A specific activity is that how much radioactivity I can load on this molecule without doing the radiolysis of this molecule that I have introduced, which is my anchor moiety. And that is very essential. And that's why we work in such a way that molecule is not perturbed, everything is maintained. And then if something has happened, then of course I'm not showing the dirt behind that. A lot of purification has to be done. That chemistry I have taken away because that will be too much to discuss today. So you can certainly, if you have gallium, you can do with this scandium. And this is what we wanted to achieve. I will take a couple of examples of chemokine receptor that we worked very recently, and that was the drive in the COVID time, that's why we want to work on chemokine receptor, because it overexpresses in many. But for me it's very essential, because when I was in UC Davis, I had designed this molecule, you can see in the end, that is called bis macrocycle, the bis cyclin, that is being used for the HIV. And even they are using for one of the molecule that is called AMD3100, quite often being used for the chemokine receptors. So I thought that is a very old chemistry I did in 93, 94. Let me change it and then think about something that COVID either get more graduated. So we took example of this one. Why I had to change this one? Because I wanted to do the copper chemistry in a different way. So that's why I took the crystal structure of the molecule that I have reflected in this microcycle in the nature, because I know that how copper binds, what is the coordination number, to multiply it, and then I took this one, reason being that the crystal structure was known, and then I joined these two molecules to load again some biological receptor, so at least we very honestly load the copper to do the conjugation chemistry to send the site where we wanted to send. Why I took this one, we look at this molecule is being exploited with the M+3, copper comes +2, +1, because it's very essential to also see that what is the electronic configuration to work around. You can see that how nicely tumor is visualized. And keeping that in view, we thought that we can produce this molecule, and now finds that a lot of bioinformatics, molecular modeling, G-score, Z-score, minimization, DFT, everything you do to find out. So we thought that also we should subject this molecule, also qualifies the molecule known in the literature, either that is being reported in the literature, falls very nicely that you get the score of -77. That's very beautiful. And then we are able to do the preclinical studies by grafting the tumor. You can see such a beautiful image of this molecule. And that gives a degree of confidence that this molecule, I can take it further for clinical application, where you can see very nicely that region with the copper half-lives, that is the twin isotope. Copper, why is beautiful? Because Jain sir was saying that copper is the one radionuclide that has got the beta positive is almost 70%, 30%, and beta negative is 70%. So it means the same nuclei, by changing the concentration, can be used for theragnostic application. So you can diagnose and you can treat with the same molecule, and half-life is 12 hours, so I can send it anywhere in the country for the clinical application. So this is the two case studies that I wanted to share, and then third case studies I don't know if I have kept, either I have removed, and that's why here it goes around that in this area for the copper, scandium, we wanted not to buy from anywhere in the world, to produce at our site with the cyclotron, to be self-reliant for the biomedical application, and to see the other people also get benefited, and that becomes the Atmanirbhar Bharat, either global Bharat that we talk about, and to see that we do the educational growth, economical growth of the country by whatever we can contribute. I will take one or two more examples, and then I will conclude, is that the last case study that I wanted to share, and then Scott is here, that he works in neuroscience, and it's my interest to see that how they respond when biochemical change taking place, either changing the different neurotransmitter, the function of different type of tasks that we perform in the time scale. So we took this beautiful radionuclide metal ions from the periodic table. They are paramagnetic in nature because of the unpaired electron we talk about, and there is no controversy that is being used every day, like contrast enhanced MRI that we talk about. And I will be very happy to know that the molecule is written in the here, and the arrow is designed by the Mishra peptide method to develop from phenylalanine, that's microcycle, and that is being used for the different type of contrast agent for imaging purpose. So what we thought that let's take one example where we want to really see that can we really see the calcium fluctuation. That is one of the neurotransmitter and the neuroscience. We always want to see that can we trap it non-invasively to see that when neurons are firing, how this circulation is taking place. So you can just first you have to see that what is the extracellular and intracellular calcium, and then after that you design the molecule to make it MR inactive. It means I have closed all the magnetic properties of this molecule. I am not allowing to enter any water molecule to change the T1 and T2. So it becomes completely 9 coordination. Again, play with the coordination chemistry. Then I change its structure completely, and I allow to interact with the calcium pools when it gets upregulated due to the some task that we perform. And then it allows to us to enter, and then water molecule that change completely from the time scale that I want to see. So it becomes 9 coordination to the 7 coordination, and then finally we try to introduce many anchor to see that they get frustrated. When molecule fluctuates, then we are able to see it. And that's why you can look at the best example of the crab, that to hold the denticity, at the same time you have got the 2 anchor that sense the molecule is fluctuating to move forward. Keeping this in view, we have designed MELI. We have many precursor we have designed, and we have to design in such a way that even if your chemistry does, they do not participate in coordination. If they participate in coordination, then entire beauty is gone. They have to be just like an anchor. When they bind with the biomolecule, they change the property completely. I can take you one example of this that I showed you in the picture, that how we introduced 2 moiety to change from 9 to 7 coordination. At the time scale that I wanted to say the function of pH, very nicely you can see that is very exponential, that change even the concentration, the function of pH. Not only this, the beauty is this, that even you can see the incremental changes with this one, and that is 150% by change in this concentration, either pH due to the metalloinotropic that we talk about, at the time scale that we want to see in the fraction of millisecond, to change the T1 and T2. And that's become very powerful for us to really visualize it and to see it in the millisecond time scale. And that's why we are able to see it by injecting in the ventricle in the monkey, that when they perform the task, that how the neurons firing, if there is any leakage of neurotransmitter or not. With this, thank you very much for your kind attention, and any question I will be very happy to take.
Very interesting talk. I knew nothing about nuclear medicine, and I feel like, wow, there is so much one can do. So my question is, the 6-year course study that you showed, and the tracer that you developed, was it based on monoclonal antibody? Can you tell the tracer with a monoclonal antibody?
Yes, the Gothenburg in United States, he owns a company that was, before it was the SysBio International. So now, before you used to do the immunotherapy, the Gothenburg started radioimmunotherapy, synergistic effect, because you have the inhibitory as well as you are loading the beta positive, either beta negative or alpha. And that is well exploited. Now is the medicine of today, the people are doing radioimmunotherapy. Most of the center are peptide radioreceptor therapy. I didn't show you because we want to be very circulation time should be very fast, because when we take the antibodies loaded with radionuclides, circulation time and half-life is very long. So it takes time, 6 hours to clear from the body. So in that time period we irradiate many organs. So what we try to do, we try to make epitopes that recognize the antibodies. So we inject the antibodies, let it bind to the tumor site, and we inject a small heptane, that is the peptide, recognize the antibodies, and you can see very nicely.
Excellent. I need to talk to you for setting up collaborations.
Baba.
Very nice topics, sir. Very informative, and like Dr. Ritu said, I do not have any expertise and knowledge in this field. I have a very quick question. The data that you have shown is a lot of data is human data, and it appears that you use investigational molecules. My question is, how do you seek the approval for human trials when there is?
That's very good question. Very good question. We are lucky. Should I say that?
Yes.
Radiopharmaceutical in the India with Tedesco considered as a cosmetics. In 1970 Act, the reason behind is that we do inject suboptimal dose. And it is not well established when we are doing the thyroid cancer treatment with iodine 131. So still we are exempted from the Tedesco to get the human clearance, but we do have institutional human ethics committee where you have to get the approval with all the data and pharmacologists. If they don't prove, you cannot go for the clinical application. So it's only called radiopharmaceutical committee, institutional radiopharmaceutical committee, where we have a judge, layman, expert, and once they approve it, we go for clinical application. But time is not too far because now people have started making money out of you, so Tedesco will come.
Thank you very much. One last question I can expect from any student. Anyway, so I would like to thank Dr. Mishra, but to the students who are sitting here, I'll tell you that to match with the frequency of Dr. Mishra, you have to consider so many things. Number one, you should be knowledgeable in radiopharmacy, nuclear pharmacy, nuclear medicine, and molecular pharmacology. These are the things which may or may not be in your syllabus, may not have been taught to you, but then there's a very interesting, and if you want to make a career in something like say nuclear medicine and nuclear pharmacy, I think these are the good options which are available to you. I thank Dr. Mishra for a wonderful lecture, and I'm inclined to say that it's very eye-opening so far as the future generations of nuclear medicine are concerned, and they are going to be really helpful to my students if they really take up this particular challenge. They must understand what is the need of the day, what is the need of tomorrow, and this is what the answer is there in terms of the theranostics and all these things what Dr. Mishra has been talking. Thank you very much.
Thank you very much, sir. Thank you very much.
I would request Dr. Rizwan Khan, Principal SKICON, to present the memento to Dr. Jain, the session chair, as well as Dr. Anil K. Mishra, the keynote speaker, as a sign of gratitude.
You see how quickly alive this converted to the glass? I love it, sir.